Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Radiation Toxicity, Considerations for Implementing Radiation Dose Limits in Radiopharmaceutical Trials. At this point, I'd like to thank Medpace who developed the content for this presentation. Medpace is a scientifically driven, global, full-service clinical contract research organization providing phase 1 through 4 clinical development services to the biotechnology, pharmaceutical, and medical device industries. Medpace's mission is to accelerate the global development of safe and effective medical therapeutics through its high science and discipline operating approach that leverages local regulatory and deep therapeutic expertise across all major areas. Now it's my pleasure to introduce you to your speakers for today's webinar. And first, I'd like to introduce you to Dr. Jess Garnaschelli, Senior Medical Director at Medical Department with Medpace. Dr. Garnaschelli is a radiation oncologist. She is a board certified by the American Board of Radiology in radiation oncology, radiation biology, and radiation physics. She brings more than 2 decades of experience in clinical research, academia, and clinical radiation oncology practice. And next, I'd like to introduce you to Dr. Stephanie Millen. She's a clinical trial manager at Medpace. Dr. Millen obtained her PhD in physiology and anatomy and genetics from the University of Oxford before joining Medpace clinical operations. Having trained initially in oncology, she now specializes in managing radiopharmaceutical trials globally, including both imaging and therapeutic compounds. And next, that's Deborah Herscher. She's a Senior Director of Clinical Trial Management at Medpace. And Ms. Herscher has more than 18 years experience in clinical research, serving as Clinical Research Associate for 2 years and managing clinical trials for 16 years. She has managed clinical trials in a variety of indications, including oncology, radiopharmaceuticals, and general medicine. And next we have James Thomas. He's the Senior Director of Regulatory Submissions with Medpace. James has a combined scientific, legal, and ethical background to conduct and oversee submissions and approvals in over 65 countries from phase 1 through 4 over the last 16 years. He interacts with regulatory authorities, government policy stakeholders across the spectrum of therapeutic indication and trial types. And next is Dr. Daphne Villewine. She's a dosimetry specialist and senior project manager at Medpace Core Labs. Dr. Villewine is a physicist with a PhD in radiophysics and medical imaging, postdoctoral training at the National Institute of Health. She is managing phase 1 through 2 radiopharmaceutical trials throughout Europe and North America. And now it's my pleasure to pass the mic over to Dr. Garnaschelli. So Dr. Garnaschelli, when you're ready, you may begin.
Thank you, Sonia, for that introduction, and thank you to our Medpace radiopharmaceutical team and the audience for joining us today. When the general public thinks of radiation toxicity, they may think of nuclear disasters such as Chernobyl, Hiroshima, Nagasaki, from which we certainly learned a lot about radiation toxicity. But in terms of radiation treatments, radiation therapy has been used for over 100 years. It is the oldest cancer therapy, and the role of radiation therapy has increased with time in treating cancer. This is due to its predictable mechanism to kill cancer cells differentially. Thus, we anticipate applications will continue to expand over the next several years. This also means we need improved ways to apply it safely on a global scale without excess toxicity to the patients we are trying to help. It's in that context that I'd like to talk about radiation toxicities, and here we have the classic picture from Hall's book of radiobiology. We must always go back to the radiobiologic principles of cancer death from radiation. Radiation causes single and double strand breaks of DNA, leading cells to apoptose or die. The efficacy is dependent on so many biologic factors, such as the big R's of radiobiology, which are repair, reoxygenation, redistribution, and repopulation, but also many other factors that I will discuss shortly. So what is the sequence of events following radiation exposure? It's important to understand the events occurring during and after irradiation of tissues and cells. So radiation can damage cells by both direct and indirect actions. The direct effects describe radiation directly causing irreparable damage to critical targets within the cell and DNA. The term indirect effects describes the situation in which radiation interacts with other molecules of the cell that are not critical targets, but are close enough to pass on this damage, typically in the form of free radicals. So this visual shows the sequence of events in cells and tissues following radiation exposure. In less than a second, energy is absorbed, hydroxyl radicals are ionized, DNA radicals are generated, then DNA is oxidized and breaks within seconds to hours, and in hours to years, cells die, apoptose, or they survive, but go on to develop mutations and carcinogenesis. Then weeks to years later, tissue effects manifest, which include fibrosis, thickening of tissues, scarring, vascular damage, and organ damage. So how do radiopharmaceutical agents impact cells? It depends on if you are employing a beta particle or an alpha particle or even electrons. So radiopharmaceutical radionuclides emit beta particles, alpha particles, or electrons. Similar to external beam radiation, each emission type can cause damage to cell DNA and organelles and generate destructive radical oxygen species, all of which can result in cell death. Given the potential for lethal damage, it's critical that these radionuclides are directed to tumors and not to normal tissues. And one must know the biodistribution of an agent to anticipate tissue toxicity and side effects. For those unaware, there are many approved agents in radiopharmaceuticals. The process required to obtain FDA approval of radiopharmaceuticals requires significant investments in production, conjugate synthesis, and validation of in vitro and in vivo performance before first-in-human studies. And these processes have been comprehensively reviewed in our other Medpace webinars. The FDA-approved radiopharmaceutical agents are outlined in this cartoon here. Agents can be categorized as either passive or active with respect to targeting. Radioactive iodine-131 is used for well-differentiated thyroid cancer, and it exploits the natural uptake of iodine in this cancer. Samarium-153 localizes to areas of bone turnover sites of osseous metastases and are available for this indication, bone metastases. Similarly, the alpha particle emitting Xofigo is approved for men with metastatic castrate-resistant prostate cancer, those with symptomatic METs, metastases limited to the bone. Zevalin and Bexar received FDA approval for patients with relapsed or refractory non-Hodgkin lymphoma after demonstrating excellent response rates. The most recent and promising iterations of radiopharmaceuticals have emerged for patients with neuroendocrine tumors and prostate cancer. SSTRs are overexpressed in neuroendocrine tumors and can be used to target these malignancies. The phase 3 NETTER-1 study showed that lutetium-177 dotatate improved progression-free survival compared with standard of care, even though it did not show improved overall survival in the final analysis. And given these results, Lutathera received FDA approval for treatments of SSTR-positive gastroenteropancreatic neuroendocrine tumors in adults. Of note, it's important to understand expected toxicities from the FDA-approved and most commonly used radiopharmaceutical therapies. Radioactive iodine, which I mentioned is indicated for thyroid cancer, is a beta particle, and toxicities are most commonly dry mouth, which is xerostomia, nausea, vomiting, transient myelosuppression. Some patients also experience radiation-induced thyroiditis, nasolacrimal duct obstruction, thyrotoxicosis, and later on, some develop secondary malignancies, pulmonary fibrosis, and hypothyroidism. Samarium-153, or quadramet, which is also a beta particle, has been reported to cause myelosuppression, nausea, vomiting, asthenia, anorexia, bone pain flare, and sometimes UTIs. Xofigo, as mentioned, used for metastatic castrate-resistant prostate cancer, is an alpha particle, and past toxicities reported include bone pain, anemia, and constipation. Lutathera, for neuroendocrine tumors, which is a beta particle, have reported side effects of nausea, vomiting, and diarrhea. When thinking about dose constraints for patients enrolled into radiopharmaceutical and radiation oncology studies, it's important to consider the most appropriate normal organ dose constraints. And the most typically thought of organs are listed here. Traditionally, radiation oncologists have learned and referred back to the classic radiation tissue toxicity tables that were created by Dr. EMANI. These tables provided partial volume radiation tolerance estimates for a few organ sites, and this was published back in 1991 in the International Journal of Radiation Oncology, Biology, and Physics, or otherwise known as the RED Journal. It reported anticipated toxicities tabulated as TD55 and TD55 for doses delivered in fractionated ways, which was 1.8 to 2 gray per day. Later, thanks to Dr. Benson and his colleagues, the EMANI tables were updated when the QUANTACT data was published, and that's the quantitative analysis of normal tissue effects in the clinic. They provided new consensus guidelines for early and late radiation effects and described this volume effect and normal tissue complication probability in terms of models, complicated models. And these were extrapolated and are now used for modern day external beam radiation therapy and are very helpful and informative to predict outcomes from external beam radiation therapy. When delivering external beam radiation therapy or radiopharmaceuticals for therapy, it's important to consider other factors that may impact radiation toxicity and a patient's inherent sensitivity. And this includes hypoxia, radioprotectors that may be used, cytotoxic chemotherapy, targeted therapies and biologics, and hyperthermia. Additionally, when enrolling patients onto a radiopharmaceutical therapy trial, it's important to consider common comorbidities. And we must weigh those comorbidities as they can predispose a patient to increased radiation sensitivity. These include scleroderma, lupus, inflammatory bowel disease, HIV, diabetes, and multiple sclerosis. Hereditary syndromes impact a patient's inherent radiosensitivity, and these include ataxia telangiectasia, Fanconi's anemia, Li-Fraumeni syndrome, among others. At Medpace, we always encourage companies who are developing radiopharmaceutical trials to consider their enrolled patients' previous radiation and cumulative radiation exposure. Our in-house dosimetrists and physicists, Daphne Valoing and I, work together to evaluate each enrolled patient's past and clinical trial dosimetry. To avoid unexpected toxicity or surprise side effects in patients previously treated with radiation prior to radiopharmaceutical trial enrollment, we encourage that the site or target radiation technique used in the past be collected. One must understand the total dose delivered, the previous modality for treatment, and we encourage sites to send a PDF treatment plan to our center for evaluation. At Medpace, we do have recommendations for interpreting previous radiation documents. Classic dose limits are derived from old external beam radiation days, and limits can be like the organ dose limits you see here. But it's important to consider we are not comparing apples to apples when we are using old limits from external beam radiation therapy days and then applying them to radiopharmaceutical limits. There are equations one must use for the conversion of different fractionation schedules or dose rates. For conversion of hypofractionated and stereotactic doses into the 2 gray fraction equivalent, one must use the EQD2 and consider both alpha-beta ratios. An alpha-beta ratio of 10 is typically used for tissues and tumors that respond early to radiation treatment, and an alpha-beta ratio of 2 is typically used for organs and tissues that respond late to radiation treatments. And I will pass the presentation over to our clinical operations team.
Hi, good morning, good afternoon, good evening. It's Deb Hersher. I'm going to start this part and then be joined by Stephanie Millen, my counterpart. I did want to emphasize, and I really can't overemphasize, how important teamwork is to managing radiopharmaceutical trials. An organized coordinator who has established and maintains the interdepartmental relationships really is a key driver to the success of these trials. We want to make sure that documentation for previous and cumulative exposure calculation is collected, as just emphasized. A good time to begin this is in the pre-screening process to make sure that we have collected all the important information to assess the patient's eligibility. We want to make sure that due consideration has been given for emergency equipment and medications to be available in the immediate environment for dosing. Now, there's multiple people involved with dosing, another key component of teamwork, because you may have a radiation oncology tech there or nuke med tech there, also a nurse. The CRC is also going to be around collecting information. So communication and good working relationships is very important. Also, keeping in mind the principles of OLERA, time, distance, and shielding, to make sure that we prevent unintended radiation exposure is important. And it's a continuing education process because, as I said, we work with teams. We know there's going to be team turnover. And so sharing that and re-educating as needed will help with the comfortability when dosing is provided. There needs to be ongoing assessment for signs of extravasation. And it is important to make sure that in the event of something like this, we've got a good plan in place and we have that authorized user in the building available to come and assist as indicated. It is important that during the dosing that we are immediately notified, whoever, for safety reasons, if it's under or overdosed by 10% of the prescribed dose. So the medical monitor and sponsor will need to know this information and then provide any kind of follow-up recommendations to the dosing team. All right, so everybody plays a role in patient safety. We never know who is going to be the first person that sees a medical record or a physical paper chart and notices that there's some kind of event that has been identified. We want to make sure that if it's a coordinator or a data coordinator or a study nurse, that we make sure that any kind of potential findings is escalated to the investigator for immediate review so that we have a good pulse on the safety of our products. As far as assessing adverse events, we want to make sure that we use open-ended questions when we are talking to patients about their response to treatment. We don't want to ask leading questions, and we want to make sure we're collecting comprehensive events that can then be assessed for the relationship by the investigator. We want to make sure that there's a thorough look-see, making sure that the EMR has been reviewed, any paper source has been reviewed since the last patient visit. We also want to make sure that we're also looking at the nuclear medicine notes during imaging studies. These are not things that maybe routinely are done. So a coordinator may need to visit departments to review those, especially since uploads to an EMR system, for example, may be delayed. And we really want to get a pulse on what is going on with the patient from a safety perspective in real time. Again, we want to make sure that when we're looking at physician notes, we're looking at the physical exam findings for symptoms of early radiation effects, and then also for late radiation effects. So as Jess had alluded to, some of the early radiation effects we would be looking for involve the skin, the mucosa, the salivary glands, and the abdomen. And then there's also indicators of bone marrow suppression that can be identified because we are doing frequent laboratory assessments on this group of patients. As far as ongoing and late effects, targeted physical exams and labs during that follow-up period is going to be important to help us identify those late radiation effects in the neurologic, pulmonary, renal, and then late bone marrow failure, which we expect to see some of those late effects. We want to make sure that the investigators are seeing the adverse event logs in real time. I recommend establishing a routine weekly meeting with the investigator. Maybe it's only a 5 to 10 minute meeting, but making sure that we've got those logs completed by the coordinator so that the PI can assess the relationship in real time, and we can get that information into the database, which everybody has access to. It is also best practice to have the SAE signed off in real time at that review, so maybe the coordinator can make sure that the EDC is pulled up as they start the conversations about relationship and take care of that and be efficient with the time, but know that the safety information has been reviewed in a timely manner. Any trends we can share proactively with the wider team. In phase 1, we're having weekly safety calls. In phase 2, we will also have safety calls at a certain cadence, at least monthly probably, just to keep everybody up to date with what is happening. I'm going to pass over now to Stephanie to continue the conversation about clinical operations.
Thank you very much, Deb. So we've walked through so far some very important points about site-level identification of radiation-related AEs. But what I wanted to touch on here is that it's very important to set up the study with mechanisms in place to keep oversight of study-level cumulative safety events and radiation toxicities, and that allows us to paint a holistic view of the safety profile. We need to ensure that when we're setting the study up, that any radiation-related toxicities are identified in a timely manner, and that the processes in place to identify and report them are as robust and as reliable as those that are already very well established for the more traditional DLTs and AEs. This allows us to be confident in our identification of study triggers, such as stopping rules or SRC, DSMB milestones, and also that any escalation or de-escalation component of the study is being carried out correctly, and that ultimately ensures the safety of our patients. In setting that up, we need to consider the additional data sources and data flows associated with the calculation and reporting of radiation-specific data. For example, adapting our ECRS to allow accurate reporting of radiation-induced events. And as Dr. Garnasheni has already highlighted, that needs to include any relevant comorbidities and historic radiation exposure as well. If we anticipate any organ toxicity identified from dosimetry reporting, then we need to consider what data is needed to feed into that modeling and also in what format it's being delivered. That data might be in a report, it might be tables, it might be listings. So how is that then integrated into the larger processes involved in safety oversight to ensure any toxicity identified is flagged to the relevant study team members? So we need to ensure when setting the study we have all these processes in place to take data from different sources and in different formats and pull them all together in a way that's informative, both for specialized team members, such as our dosimetrists and our SMC members, but also our non-specialist study team members. So that might include other members of the study team, and also a number of site staff that quite simply, particularly if they work on, may not have come across this data before. Once we have that all sent up and we're fully informed, we can then review the emerging safety profile comprehensively and identify any actions that might be required at a study level. So the processes for AE reporting are well established. So what I'm going to focus on here is the consideration for setting up dosimetry specifically from an operational perspective. And there isn't a set way of doing this, but more a number of considerations that are all interlinked and dependent on each other, and that all needs to feed into the final strategy. And I'll speak through this generally, but also using an SRC meeting as an example to illustrate. So firstly, we need to consider our data requirements and what data points we need to collect from our patients. Different dosimetry modeling might require different inputs, and that might encompass qualification data, it might be phantom scans, calibration data, different imaging, blood and urine data, there's a lot of different information that can go into this model. And the different optimal time points for that data will also depend on the isotope and the drug administered. So it's very important to engage dosimetrists early on to ensure that they can work with the schedule of events that's planned in the protocol, and also that the assessments planned give them everything they need. We also need to consider the quality of the data that we want to work with and the level of cleaning and verification that will be required before dosimetry calculation starts. In a perfect world, we'd have clean data straight away, but pragmatically, that isn't always possible. So we need to weigh the risks and benefits of the different opportunities, also knowing that dosimetry calculations can be very lengthy in time, but also that the results might be used to make some very critical study decisions, such as dose escalations. Let's also consider the data that will come out from the dosimetry, because there's no point in having a lot of data if people can't understand and interpret it. So will it be presented in Word format in a report in a PDF? Will it be a table or graph or listings? If it's a Word report, how are we going to extrapolate DLTs and pull them out from that and quantify them? Additionally, is the format going to be understandable for our non-dosimetry experts and SRC members, or will it need any manipulation or interpretation? And if it does, who will perform this and what are the timelines for doing so? So we need to ensure we're allocating consideration to both the data inputs and the data outputs. In parallel with that, we also need to consider timelines. So what is the data needed for and how quickly is it needed? We know that dosimetry calculations truncate time. So are we going to compress timelines elsewhere, or is it allowable for the end result timelines to be pushed back a little bit? If it's for an SRC meeting, for example, when will the meeting occur? How long do attendees need to review the data ahead of that meeting? All of these considerations need to be taken in together. And once we establish some of those timelines, it might then loop back into the data strategy. So if we're very time constrained, it might simply not be possible to have clean data. So you can see we're not looking at a process that A leads to B leads to C, but more a lot of considerations that feed into the same developmental strategy. And once we've got an outline of our core activities, we can start filling out all of the different processes that feed into it and looking at all of the contributing factors. So continuing the same example, once we've set our timelines for our SRC committee, we then need to schedule our data transfers. That requires data to be entered within a particular timescale. So taking a step back, we then need to look at the communication pathways to the site. And ultimately, how is the study coordinator or the data coordinator knowing what data to input? For example, do they only need critical data, or is everything needed? When does it need to be input by? Taking one more step back, so taking a further step, how is that data actually making its way to them? So the study coordinator knows what data they need to put in and in what timeline. That ensures it gets to the dosimetrist and the SMC on time. But how is that data actually getting to the coordinator? Because that data might come from oncology, it might come from nuclear medicine, it might come from the radiopharmacy. So what processes can we put in place to ensure that we're lining ourselves up for success for that whole process involved in this? And that might then feed into study documents such as the monitoring plan, for example, looking at our monitoring strategy and the activities conducted during the SIV to know how that source data is transferred and who is responsible. So you can see that even building a dosimetry process for an SRC has to incorporate a lot of different factors. And this will apply to numerous different set of activities, not limited to an SRC, but might also include our processes for DLT identification oversight, any data visualization we might want, interim analyses, and many, many more things. This was just one example, but dosimetry processes need to be considered in detail from the start of the study and engaging all parties early on to ensure that they're seamlessly integrated and embedded into the study execution. And that leaves me nicely to hand over to Daphne, who will now present some radiation dosimetry concepts.
Thank you, Stephanie. As was already discussed by Jess earlier, it is very important to consider the dose limits in radiopharmaceutical clinical trials. So for this, we can calculate the cumulative absorbed dose to organs and we can also estimate maximum activity to administer once we have performed the dosimetry calculations for one cycle already. So in order to fully understand what all this means, I will start with some background on the dosimetry calculations for radiopharmaceutical studies. So in the dosimetry of radiopharmaceuticals, we calculate the absorbed dose following a specific formalism that was developed by the MERT committee, and that expresses the absorbed dose to a target region as the product of two factors. First, the time integrated activity in a source region, which corresponds to the total number of disintegration that happened in that region of interest, which can be an organ or a tumor. And secondly, the S value, which corresponds to the absorbed dose to a target region for one disintegration that happened in the source region. So some target regions are also source regions when the energy deposition from the emitted particles is very local. We call that the self-absorption, but some target regions are only receiving the energy of particles that were emitted somewhere else, which we call the cross-irradiation. Now, to apply this formalism, we can follow several main steps that altogether form what we call the dosimetry chain. For image-based dosimetry, the first step is the image processing, for example, the image registration between the anatomical and the functional image, but also between different time points. The second step is the segmentation, during which we contour the different regions of interest on the images. Then the uptake is quantified in these regions on the images acquired at all the time points. The fourth step is to calculate the area under the time activity curve, and for this to perform a curve fitting of this time activity curve. And finally, the last step is to take into account the transport of particles within the body to obtain the absorbed doses to all the target regions. So one consideration we have in internal dosimetry for that last step is, should we perform these calculations using the actual patient images, which is called patient-specific dosimetry, or should we use instead some representations of the human body that we call anthropomorphic models? So you can see here different anthropomorphic models. They evolved from the stylized versions in the 1980s that were not very realistic, to voxel models and now hybrid models that are way more realistic and representative of the human body. It is recommended to perform patient-specific dosimetry for therapeutic compounds rather than model-based, since it accounts for intrapatient anatomical variability and way better tumor dosimetry. But patient-specific dosimetry involves complex in-house developments for applications in clinical trials, especially for emerging radionuclides. Therefore, model-based dosimetry is sometimes the most appropriate option. Now, let's get back to the use of the dose limits. Once the full dosimetry calculations have been performed, we can obtain the cumulative absorbed dose to a given organ over all the cycles of a treatment, and also preferably over all previous types of radiation therapy. This way, we can verify that these cumulative absorbed doses to some specific organs that are considered organs at risk are kept under some dose limits. When we talk about dose limits, we talk about the absorbed doses and not the dose in terms of quantity of product administered. These dose limits have been established from external radiotherapy, which may be a limitation to keep in mind, and they are available from the literature as discussed earlier by Dr. Guarnaschelli, such as, for example, this paper for Marx et al., which presents a good summary. This means that if the cumulative absorbed dose to the kidneys is superior to 20 grays, there is a risk for toxicity, for example. Same thing for the liver over 31 gray, etc. So now, how can we monitor that we are going to exceed these limits? So we can use the dosimetry results from a first cycle to estimate the maximum activity to administer over the remaining cycles. So here is a hypothetical example with dummy data. Let's say that for this study, we have calculated the absorbed dose coefficients to this list of target organs. From these values and the known dose limits, we can calculate the corresponding maximum administered activities for each target organ. Here, in this case, the pancreas and the spleen are not organs at risk, but all the others are. And the target organ that has the lowest maximum administered activity here is the red bone marrow. This means that in this case, the limiting organ is a red bone marrow, and that the total administered activity not to exceed over the different cycles is 45 megabecquerel. I will now move to more regulatory aspects and discuss the use of radiation exposure data in the informed consent forms. So in these ICFs, we refer sometimes to the effective dose, but this quantity should be used with a lot of care. I listed here the different inappropriate use. First, the effective dose should not be used to predict cancer risk among exposed patients or persons, and in retrospective investigations of individual exposure and risk. In clinics, we should not use it for treatment decisions. And lastly, we should not use it to assess the radiation dose to medical patients. As was reminded by Fisher and Fahy, it is the absorbed dose that is the relevant quantity for evaluating radiation dose to organs and tissues from exposure to external radiation and internal radionuclides in the workplace, the environment, and in medical applications. Now, for the informed consent forms, when should we use it or not, and what should be the language? Well, the main question is the situation involving therapeutic levels of activity or not? If yes, then it is not appropriate to use the effective dose and to make a comparison with the natural background exposure. What should be discussed instead in the ICF is the absorbed doses to the individual organs. Then if the answer is no, and we are looking at diagnostic levels of activity, then risks can be considered either minimal or acceptable. The effective dose can be used and the comparison to natural background can be appropriate. Both may or may not be appropriate depending on the level of exposure. The absorbed doses to individual organs may have to be provided as well. So here a proper evaluation needs to be made, and this is something that actually Medpace can help with. So finally, here is one example of what we've just seen with the ME data again. So this ICF mentions a therapeutic radiopharmaceutical. However, we can see that the effective dose is reported with a very high value of 620 millisievert. We also see a comparison with natural background exposure and medical exposure. And from what we've just seen before, we know that the language should be modified in this ICF to remove mentions to the effective dose and the natural background, and to add information on the absorbed doses to individual organs. So this is it for this section, and I will now hand this over to James Thomas for the regulatory considerations.
Wonderful. Thank you, Daphne. I particularly appreciate that tangible example. I suppose I'd like to carry on that theme from a regulatory perspective. So where it's looking at the regulations, the guidance, the best practice, be it global, regional, country, or site-specific, and that practical, continuing on that practical application. And I think that's typified in presenting quite complex information like you've just described. And the key for me is balancing how the information is presented. We've just heard about how Medpace's knowledgeable team members can help support the calculation of the data and having that right technical data available. And though it's going that next step and putting it into the right context to present it to the right audience. So balancing the technical and the accessibility of that information. So on the one hand of that scale, it is having that data, the ability to have the right information to present to regulators, ethics committees or IRBs, dedicated radiopharmaceutical review bodies, who are going to have maybe their own perspectives and focus on certain aspects compared to participants who may look at it from a very different angle. So that technical information being available to include right information in detailed application forms, but to have the right summary of information so it's presented in the right way, supports and facilitates the review of that information from those review bodies. And the cover letter is a key tool for that, and it's about adding value in our experience of presenting the information in the right way, taking that technical information, but then balancing it with the right context for the right situation. An ethics committee, for example, may be looking at it more from the participant perspective than a regulator is going to, perhaps. So how do we maximize that engagement with the audience? As I said, balancing the right technical information in the right places versus having the right context for participants and what it actually means for them. And I'd like to maybe go into a little bit more detail around some of that patient-facing material and how we can help support that, and some of the differences we see around the world. So I guess there are three aspects to ensuring that there's relevant, helpful information. One is providing enough educational material for potential participants. The second one is to ensure there's appropriate consent and truly informed consent over quite technical information, and looking at that, maybe having some materials that the participants can access and refer back to during the course of the trial. So that first one, that education, it helps support and engage with participants and helps the site to allow that to happen by helping making them aware about what it might involve, helping ensure that they understand some of this complicated information, what it might mean in practice for them, and then starting to build that more into the consent, whether that's risk language, which I think is a key area, as we've just heard, in the toxicity in particular. What's the impact upon the body? What does it mean for that person? Numbers and that technical information is incredibly helpful, but we have to be able to make it understandable for those individuals who are consenting. It has to be in those lay terms. And I think not just making it understandable, but putting it into the right context becomes key also. So context and impact. What's it going to mean for me if I'm participating in a clinical trial? So balancing that information with what are the regulatory requirements, whether it's a legal liability perspective, whether it's ensuring that we're complying with specific language that regulated bodies want to see in there. But that balance is, I guess, the main theme. And that can include things around what it means for you after the trial or for the duration of the trial. And I think one thing that we found particularly successful perhaps is having a separate precautions document that really allows us to go into a little bit more detail and have a useful reference for participants throughout the course of the study. So an example of that is what type of information would we include in here? So again, just a couple of examples, very practical information. Who they avoid contact with if they are potentially exposing to radiation information. How to help manage some of that risk, showering daily, for example. What does it mean for them in reality? If they have to avoid contact with their partner, their family, their close friends, that can be quite isolating for the individual. And it's important to ensure, again, true informed consent for participants coming into the study. And they may need to refer back to these throughout the course of the trial. And I think that's the balance that I talked about, that technical information, having that available, and our regulatory submissions team here at Medpace cover the regulatory authorities and the ethics committees, which allows us to be able to understand the two different perspectives, perhaps, a little bit more, and allow the right information to be presented in the right way. So lots of different tools and techniques that can be used, and I just wanted to highlight a few of those today. I'd like to pass over now to Dr. Ganshali to help conclude.
Thank you so much. So the key takeaway points that we would like our audience to take away today are that patients' cumulative radiation exposure, the concomitant medications that they are on, their medical comorbidities, and hereditary syndromes will impact their inherent radiation sensitivity and should be considered. When setting up a study, we must consider the data flow and processes involved in the evaluation of data that's required to identify radiation toxicity risks. In addition to the dosimetry while on the study, patients' cumulative absorbed dose to organs at risk, and the maximum activity to administer based on dosimetry results from the first cycle are really important assessments. Use of effective dose and comparison to natural background exposure in the informed consent of therapeutic radiopharmaceuticals is not appropriate. And it's important to balance information to ensure that it is pertinent and in an appropriate context and not excessive. And that concludes our presentation, and I will pass it back over to Sonia. Thank you so much to the audience for listening today.
Thank you very much for that, Dr. Gurnashelli, James, Dr. Millen, Dr. Villawine, and Ms. Herscher for that very insightful presentation. I hope everyone enjoyed it. Now I would like to invite our audience to continue sending in their questions or comments right now using the questions window for the Q&A portion of the webinar. I've already received a bunch of questions while you were speaking, so I'm going to start with those. And it looks like our first question may be for James. And the question this audience member is asking, are there any country-specific requirements regarding radiation risks that need to be considered in ICF and patient materials development? James?
Great, thank you. Wonderful question. Yes, is the answer. So I suppose we definitely see that. We've worked on radiopharmaceutical studies across over 20 countries, across various regions, North America, Latin America, Asia Pacific, Europe, Middle East, and there can be variation around here. Two examples perhaps that come to mind would be maybe looking at comparing and contrasting Australia with perhaps the UK. So in Australia, you'll have each site radiation officer performing a review of the informed consent form. The process is maybe driving here where there's a site level review, and that leads to maybe site specific aspects being discussed and incorporated into the consent forms, compared to the UK, which is more of a country-led process. And it's perhaps quite well established where there are specific bodies with the HRA's radiation assurance, but also RSAC as a review body, where there's an expectation around specific and mandatory wording to be included in the informed consent form. So in summary, I guess the answer is yes, in simple form, and those are kind of two examples of perhaps polar examples of where that can come up.
Oh, thank you very much, James, for answering that question, for providing those samples. Okay, now I'm going to go to the next question, and this one looks like it might be for Dr. Gurnashelli. This audience member is asking, what are the most important hereditary syndromes to consider when evaluating patients for radiation treatment? That's a great question. Dr. Gurnashelli?
Okay, that's a great question. So there have been many studies of genetic syndromes that are associated with mutations in genes and in DNA repair pathways. And those studies helped us gain insight into the human variations in radiosensitivity. I would say ataxia-telangiectasia, which is associated with mutations in ATM, was one of the first syndromes that was shown to have clinical and even possibly life-threatening radiation toxicity and inherent cellular radiosensitivity. But there are many other syndromes that have been identified. There are syndromes associated with genomic instability and a high risk of radiation-induced cancers once patients are exposed to radiation. And an example of that would be EMANI, which is TP53. And another one is retinoblastoma. But in those syndromes, the clinical radiosensitivity are not really seen. It's just the inherent risk for developing radiation-induced cancers after exposure. Thank you.
Thank you very much for that detailed answer. Now let's go to our next question here. This audience member is asking, how is the effective dose different from the absorbed dose, and when do you actually use it? Who would like to answer that one?
I can answer this. This
Thank
Daphne.
you. Thank you.
Yes, I did not get in much details about this. So the effective dose is actually calculated using the absorbed doses to the different target organs. So it's a physical quantity. It's been defined by the ICRP, which is the International Commission on Radiological Protection, and it is to be used for radiation safety purposes. So we use the absorbed doses to calculate it, and we apply some weighting factors that account for well, the differences of impact between different types of radiations, for example, and also the differences of radiosensitivity between organs. So that is, I think, for the first part of the question. And to answer the second part on when do we actually use the effective dose, well, we mainly use it for radiation protection of radiation workers. As I said earlier, it's for radiation safety purposes. So, for example, for nuclear medicine technologies protection in our situation. But we can also use it to compare typical radiation doses from different diagnostic procedures. And well, to some extent, and in under some specific conditions, we can use it in the ICFs, as we have just seen. I hope that answers.
Yes, thank you so much. Thank you very much for that detailed answer there. Okay, I'm going to jump to the next question now. How is e-consent allowing a targeted explanation of the complex matter according to the knowledge level of various patients? Who would like to answer that one?
Yeah, thanks. James here. I think I'll start on that one. I mean, so e-consents are much more prevalent and prominent. They're established. I see no specific issue in using e-consent. I suppose the point that we're making is that I think I have seen some enhancements that can be made around the information and how it's presented in the document in the first place. Now that e-consent doesn't replace or remove that discussion and conversation that's going to happen between potential participant and the site. But I think that ensuring the information is in the right context and is very clear. And if you're doing that, it's resonating with the individual, whether that's an e-consent or not. But just to be clear, of course, this is highly technical information that the participants may and should have questions and need to be able to answer those and have access to do that. I don't see e-consent as replacing that. I just see that as a different way of that happening.
Okay, thank you very much, James. I'm going to squeeze in one last question before we close out. This audience member is asking, most regulatory agencies are very strict and conservative on the cumulative absorbed dose limit they allow in phase 1 to 2 trials, especially with new radiopharmaceuticals. From your experience, how best to address this in phase 1/2 IND and CTA?
Great. James here again. I think I'll address that one here too. We could talk at great length around this, so excuse me for being brief. I think that one of the first things, it's about looking at the clinical development plan and setting up that sort of from a true regulatory perspective at an early stage and having dialogue with regulators. I think, again, context becomes really important here. If it's something innovative, it's something new, which very well or very likely to be at this early stage of development, all the more reason to engage with regulators at an early stage. I know we focused today a lot upon how to present the right information in the calculations. I think the question's a really good one about engaging very early with regulators, presenting the information in the right context again. I know I focused a lot on the accessibility of it, but I'm going to say accessibility for the right audience again, the regulators, potentially even engaging with some of the radiation bodies at quite an early stage. That's something that I've seen and we've done at times, and that's something that can have great value as well. So forgive me for being brief, but that's one highlight I would provide.
Oh, well, thank you so much, James, for answering that. Well, thank you very much for those answers. We have reached the end of the Q&A portion of the webinar. If we couldn't attend to your questions, the team at Medpace will follow up with you after this presentation. And if you have any further questions, please direct them to the email address that you see on your screen, info@medpace.com.